planned for today. As Jackie said, we'll have a little intro over the most common eDNA tools, what the data generation process is like, not in too much detail. There's plenty of good resources for reading about what is PCR. We're not going to cover that. What a data observation is with eDNA and how that differs a little bit and is similar in some ways to other methods, and hopefully hold people's hand on the way towards a more probabilistic understanding of their data, which is inherent in a lot of methods, but is particularly problematic, I guess, for eDNA when we are used to more familiar methods. And it's entirely natural that eDNA as a fairly new toolkit is subject to a lot more scrutiny. We want to help people better understand that so that they can use it more usefully. And then, as Jackie said, I think one of the most important aims of these kind of webinars is just to hear from you. So I'll try and leave a really good chunk, maybe even half of the time, just for Q&A at the end, so that even if you didn't get much out of the slides, you can get a question answered. And we will, of course, try and follow up on anything that we don't get to in the allotted time. So with that, the two main tools, I guess, that most people are familiar with and that I'm going to dwell on today are really sort of rapid targeted tests like quantitative PCR, although rapid field-based, sometimes isothermal tests like LAMP and RPA are gaining some really cool uses when the time of result is really, really important. But mostly I'll be talking about qPCR today. The same sort of results can apply to outputs of isothermal tests. And then the slower and more general, much more powerful approach of metabarcoding, which is PCR coupled with high throughput sequencing. So without dwelling on what PCR is, in general, it is as exponential amplification of a small piece of DNA unique to a species. And that sounds pretty simple, right? And pretty straightforward. And indeed, it is a lot simpler than metabarcoding, as we'll go into. But even so, it is a process which is very much dependent upon the data at a given point in time that is used to assemble an assay. So that is true, but it requires representative genetic data for the target species to make this assay. And even defining what is representative for a given target species may be difficult, and that itself requires data. We require representative genetic data for off-target species, and so that assumes some knowledge of the phylogeny and assumes also that we can define what is relevant off-target species. So it's based on spatial occurrence data. We may be concerned about a particular species that is in the same genus and co-occurs closely, as far as we know, with the target species. That makes things more complicated. But it is very much a data-dependent process. And so these are some of the main steps that we go through in designing assays. We identify potentially confounding taxa. This can be very easy in some cases, like for platypus most famously, or a Tasmanian tiger. Can be very much harder in the case of, depending on the vagaries of population genetics and phylogeny. So we identify these potentially confounding taxa. We collect data, either explicitly for the purpose of assay generation. That means we will source reference tissue from the species in the particular area in which we are working. We will strive to make that as comprehensive as possible, although it can be quite difficult. We will align that sequence data for a particular locus. So that's, again, another decision. We will have a sort of a suite of loci that we will go to that we know are often reliable targets for our assay at a given phylogenetic distance. We will align that. We will design primers and probes, and then we will begin the process of testing that. So we'll often design multiple primers and probes, multiple assays. We will check that against reference tissue and synthetic DNA if required, if we are unable to source reference tissue. and go through all of the technical hoops to make sure that assays are performing efficiently and consistently, and determine their limits of detection and limits of quantification, etc. At which point we can then test in an appropriate experimental context with controls, eDNA samples. And so from there, we arrive at something that we would consider to be a validated eDNA assay. So very data dependent process. And from that, it follows that all assays are certainly not created equal. So this is a useful hierarchy of validation from Bettina Thallinger et al., which we use some sort of variant of internally to sort of qualify our assays. And if you are using qPCR, and if we or others have designed qPCR assays for you, you should be familiar, I hope, with some of these steps, and you should be able to ask questions about what is my validation level, how portable is this assay, how efficient is it, because this process of validation is an ongoing process, obviously. Something that is valid today in a particular project area may not be valid outside that project area, and it may not be valid in a year's time if new data comes along. You can work with the best data that you have available to you at a given time. But it's something you should be thinking about when you are deploying a qPCR assay. What efforts have been made to go through to validate this assay, and so that you can be confident of the results, because there is enough uncertainty in the results of a well-performing assay without having complications from difficult assays. So for qPCR, really, it's a relatively simple problem for good species. And qPCR can be really highly sensitive and highly quantitative. For some applications, quantifying bacterial pathogens, you can express the results in copies per unit of, say, filtered water or other substrates, which can be quite useful. If time to result is really critical, then these kind of tools can be deployed in a field context along with other assays. But for many applications, it's fairly quick. We typically turn results around for routine assays in 1 to 2 weeks, and really less than 2 of that is time spent actually in the lab. So there is room for improvement with us and other service providers in really shrinking that turnaround time. Assays can be made well or not so well, really, depending on the data and processes behind them. So I would like you to take that from this, if it's not already obvious to you, and their usefulness may change through time and space. And so ongoing validation are all really important. The problem gets a little bit more complicated when you turn to metabarcoding, which is based on PCR, and so in theory is exponential, but that varies. So you get sort of exponential-ish amplification of a small select piece of DNA that is unique-ish to each species, hopefully within a targeted group of species. So these assays can be broad. There's kind of a blur between multiplexed qPCR assays where you might be targeting a few groups, less than 10 probably. You can have relatively specific metabarcoding assays sometimes, although more generally their use is in broad scale monitoring where you don't really know what you will be finding, perhaps in a biosecurity context or a biodiversity and conservation context where you are less perhaps interested in the attacks you're finding and in other higher order metrics derived from this data. So the process here is somewhat similar initially. Of course, you will collect samples, you will extract DNA, you will amplify them by PCR, and then you will sequence it. So the difference, of course, between qPCR and metabarcoding, the main one is that you resolve always the sequences that you are amplifying. And then you go through a process of similarity clustering and assigning taxonomic labels to a database. So You again require representative genetic data for the target group, representative genetic data for the off-target species based on spatial occurrence data, preferably. This is not always available. We're quite lucky in Australia to have a quite good historical monitoring data for at least for metazoans, but that is certainly not always available. And then, of course, the database matching taxonomic labels to DNA sequences. So this is really the main, one of the main benefits of genetic monitoring tools, in that we no longer need to rely on continual input of expert taxonomic advice. We just need it written down once in a database, hopefully correctly, for all to use. And this is certainly an ongoing process. So metabarcoding, yes, optimized for detection of many potentially unknown species. The outputs in this case are not copies per unit substrate, or even thresholded copies as they are for qPCR. We'll often threshold to do just binary detected or non-detected, depending upon the project aims. In this case, for metabarcoding, you will often receive counts of amplified DNA molecules per sample. And then for reasons which many researchers have gone into in depth, and essentially due to the bias in PCR amplification, that there is not linear exponential amplification of all molecules in there. A more commonly encountered and more robust metric is the proportions of reads for a given sample, which makes it a bit more comparable across samples. Although, of course, there is the absolute counts have some useful information. It's very different to have 1 million reads for something than it is to have 100, which is going, and the proportions are obviously going to scale with the total read depth. So there's information both in the number of read counts you get. We know this has partial correlation with the amount of DNA in the inputs, which itself, in some circumstances, can have partial correlation with the abundance of the organism. But there are, of course, a huge number of caveats to that, and a large body of research showing when that works and when that doesn't. And then assay design and optimization requires, again, ongoing validation. A lot of the assays that are in use today are dependent upon the data that was available to them at the time. And so this is reference databases are being filled at a really rapid rate, and so that at least opens the possibility for improvement of assays that are in use today, and the generation of new ones that may be more or less specific to target groups. And finally, where possible, ground truthing using known species distributions can be incredibly important for metabarcoding data because of the size of reference databases, and sometimes incompleteness or the errors that are within them. Using known species distributions, for instance, for fish and vertebrates, that works quite well in Australia. Really can be very useful, at least to throw up things that are potentially interesting or potentially wrong. So that will enable you to improve your resolution and also to focus in on things that are, again, interesting or sometimes wrong, because there are many ways in which taxonomic assignment can go wrong due to gaps in the database, unknowns or even known unknowns. So at this point, this is really just the sort of a technological, I guess, introduction to the main tools before we get on to interpretation, and we could stop quickly for a few questions here.
Thanks, Luke. We've had a few questions come in. So the first one is, what other substrates can be used? Filtered water is obvious. Scats, claw marks, what other options are there for terrestrial species?
Oh, well, the list is really only limited by imagination. The common ones are the ones you mentioned. So obviously water, actual samples of an animal, if that's feathers, fur, secretions, scats, that kind of stuff. just sampling terrestrial environments. For instance, if you can use some ecology and some knowledge of that species to really target something, it sounds like you're in a sort of targeted species context, then sampling likely habitats is really useful. That will enrich the usefulness of your data. Sampling things that they are maybe eating, whether that's bones. We were able to get predator identifications from prey items, or from plants that things may be browsing on. Really, the list is endless, but it really depends on the effort that you're able to go to into sampling, of course. So that's very much a contingent kind of answer depending on the target.
So another one is for field validation that was mentioned just now. Is it considered as in situ validation? I'm quite confused with the term sometimes.
I'm not sure what you mean by in situ validation. So the sort of the last step in the validation hierarchy is to test things in a large scale field context. Having known the ideal performance of an assay, you take it to the field, you implement a monitoring or a snapshot sort of plan with appropriate controls. And so the validation there would be testing on hopefully a subset of sites that you are very confident have the target species. You can still, of course, get negative detections from sites where you even see a fish, but you will have some confidence that those positive control sites should be enriched. And then you will have appropriate laboratory controls inside as well. So I think it's partly a question about field practice. You can obviously not always get a 100% positive sample from the field. If you can, that's fantastic. But as long as you have the appropriate positive and negative controls implemented in the field plan and in the laboratory plan, you should be confident that that assay is functioning.
Right, we might do one more in this little break. Can you talk more about the correlation between number of reads and species abundance? What we know and what we don't know yet about this topic.
Right. I could talk for a long time on this, but again, it's an answer that's quite contingent on the context, right? So there's a lot of mesocosm studies that have gone into this relationship and often, not always, found a relationship between abundance and reads in a metabarcoding or a qPCR context. But I mean, you can imagine, and you don't have to imagine, you can read about it, that these things vary with everything you can imagine. So the species in itself, its size and its shedding rate, so therefore its behavior, which will vary seasonally as well as environmentally. pH of water, for instance, all of these things change degradation rates as well as and shedding rates. So it's kind of inputs and outputs. So it's enormously complex, but at the same time, averaged over many taxa and many different contexts, there is a positive relationship. There are things we could do in the lab to improve that. So for instance, qPCR is obviously more quantitative than metabarcoding, though it cannot be used everywhere. And then there are cases where it really depends on your question. Yeah. I could waffle on for quite a long time about this. Beyond saying there is a generally positive relationship, it requires more work, frankly, if you want to use that information. If you want to get to species abundance, there are other things you can do. You don't need to use read counts, for instance, and then I'll talk a little bit about this in the next section. The more robust plan is to implement this in an occupancy detection and modeling sort of framework, a distribution model essentially, where you can get to a better approximation of abundance. But this works in some cases in sort of fisheries management, where you have a very controlled context and you're only looking at a relatively narrow suite of organisms. This can work quite well. I was seeing really encouraging data for monitoring in the holds of fisheries vessels, for instance. So in particular contexts, if you do the work and you show that it is possible for your problem, your sampling method, your analysis method, and your targets, I think it's possible to get that information. You would never attempt it on, say, a really, really broad metabarcoding assay, of course, where you're comparing taxa across the full tree of life.
All right, we might leave it there for now, but thank you for your questions so far. I'll let Luke continue and then we'll jump back into Q&A at the end.
Yes, we certainly will. I better hurry up. So what is an eDNA observation and how does it differ from other observations? Well, really it's a sample from a sample from a sample from a sample, and there may be other levels of variation there. And so alternatively, that's a result from a technical replicate of an assay run on some amount of DNA from a larger extraction that you have taken from a sample in a particular place at a particular time, right? So this can be a bit scary, although many of these things are common to any observational technique. Throwing a net in a river is taking a sample, and it is not a presence-absence result. It is a detection, non-detection result. And so while the eDNA technologies themselves will certainly continue to improve and make these things better, and not least cheaper, so that we can actually do better replication. Really, right now, the antidote to this sort of complexity and uncertainty is not new. It's knowledge, it's experimental design, and statistics. So I wanted to spend a couple of slides on sort of, I think in some contexts, it's not always relevant for snapshots, but the general aim, I think, for eDNA in a large-scale context is moving to a more probabilistic interpretation of the data, where you don't actually need to rely on any one observation critically, and if you do, then you probably shouldn't be doing just eDNA. You can compare that with other methods. So Josh touched on this a bit last time, and so I'm going to move through it pretty quickly, but you will implement a sampling design. You will think about the aims that you are trying to achieve before you collect any data. You'll think about the level of replication that you can achieve, and you will design a monitoring plan or a snapshot plan, a detection plan. So you will come up with a number of sites. You will ideally sample through time, or you may have spatial replicates, and you will run them through your assay and to record detection or non-detection, which, as I said, is often called presence-absence. It's a bit wrong. And with the assumption that there are going to be no changes during your sampling season in occupancy, you can then map out your results. So here's a partial result of detections and non-detections on a blob. And you may get a number of hits from your observations. And so what do you do with those hits? What about the ones where you have negatives? You can have false negatives, you can have false positives, of course. No laboratory or field protocol is completely bulletproof. We implement a ton of controls of different types in the lab and molecular tricks to make sure that false positives are minimized, or we can at least detect them, but no one would ever claim to be perfect. What do you do with these negatives? And ideally, you should be thinking about fitting a model where you have some probability of occupancy by a given species. This can be a single species, it can be applied in a multi-species context. And the converse, the probability site is empty, is 1-, and you will have some detectability. Ideally, you will have generated this number from an assay. We have done this for a number of species. It's not a trivial exercise, and so in practice, we'll often borrow data, say, across small fishes or mammals or something like that. So the amount of this data will improve through time, where you can say, I have a detectability for my target species based on some amount of data. In the meantime, it's one of many assumptions that go into this kind of modeling. We have a probability that you can detect something given that it's there, given a particular sampling regime. If I take X number of samples and do replicates in the lab, my assay will detect it at what percentage of time. And then you can fit models. There are a ton of mature packages in various statistical frameworks for doing these kind of models. And you can then estimate the occupancy of your species across your sites and look at any other particular covariates that you might be interested in, environmental covariates, etc. So, for instance, for a given history of observations, you'll compute a likelihood, which is just the product of the probabilities for this particular series of observations across your site. And then you will solve a likelihood service to arrive at some parameter estimates in either a maximum likelihood or a Bayesian framework, as we more commonly do. And so this, I understand that this is really only targeted at a particular kind of eDNA problem. But I think the thinking about what is an eDNA detection and the probabilities that are involved in that can be very useful. And it all really depends on, of course, what you are trying to do. So I would encourage everyone who has not already read it. I mean, we in the eDNA field are particularly fond of an article by someone outside of the eDNA field, initially John Darling from the US EPA, where he sort of argues that there are fewer differences between eDNA and traditional monitoring than people would often argue, but also that it needs to be evaluated closely and as one of many tools in a monitoring framework. So you should always kind of think about this decision tree before you embark on a project, whether that uses eDNA or other methods, and think about the consequences of what detections mean. And it may be that eDNA is the only option for you, or it may be that it is one of multiple, or it may be that eDNA is coupled, it uses a sort of pre-screen, as it often is now in a biosecurity context, where it is a relatively cheap way of assaying large numbers of species quickly, which can then be followed up with other methods should anything of concern be thrown up. And so, I mean, yeah, this shift from binary presence absence to probabilities is not really always a comfortable one, but it's one that we think is necessary if you're going to be using eDNA, and even if you are not, you should be thinking about it. So I am going to stop there.